Good morning, one and all. Am I audible? Hello?
Yes, sir.
Right. 6th unit seed treatment. So, why should we treat the seeds? Treatment. Conduct the treatment to the seeds. In what purpose? In what purpose
Yes,
we treatment?
for the shelf life,
Yes.
extend the shelf life, or to
Improve
give the
shelf
best
life and
crop.
production.
Yes,
Improve production and improve germination capacity, viability capacity. These are all carried out by if we remove the pathogens, right? So in treatment, we should remove the pathogens. This we should remove with the treatment carried out without some chemicals and some of the instrument. The chemicals may be fungicides. The name itself given fungicides means which kills fungicides, which kills fungus, and bactericides, not antibiotic. In our book given antibiotic. Antibiotic may be fungicide, may be bactericide. They are all comes under the antibiotic. So inside the bacteria given kill bacteria. The kills the bacteria, the chemical type is bactericide. So fungicide, side means killing. Fungicide, the fungi kills the fungus. Bactericide kills the bacteria. Nematocide kills the nematodes. With different formulations including the water, dispersible powder, dust or slurries are used to control specific pathogen. So based on that, actually, the apply of this chemical two type. Apply of the chemicals in chemical treatments. Just wait. Chemical treatment. Chemical treatment. First of all, protectant. Second one, therapeutant. So the chemicals, the fungicide kills fungus, bactericide, bacteria, nematocides, nematodes. Right? Whether the fungicides or bactericide and nematocide, we apply on the crops in two methods. One is the protectant and therapeutant. If we say protectants, the freer the infection, the applied the crop field, freer the infection before formation. That means before attack the pathogen, before attack any pathogen, we apply chemicals. Apply chemicals. Apply chemicals. Chemicals may be bactericide, fungicide, nematocides. Apply chemicals before infection. Here before infection. Apply chemicals. Any chemicals before the infection of the pathogen. That means before getting any disease, the prior we apply this chemical. These chemical called as protectant. Examples of the protectant here the given captan, therum, organomercurial, dithene, M15, gerum, phabum, they are all. And second one is theraptant. Theraptant chemicals, the opposite. These chemicals, these chemicals apply after infection. That means we applied after infection. After infection of the pathogen. Already pathogen attack and also it's caused the disease also. Now we apply theraptant. So the example carabdogenum, Bobstal, oxazine, binomil. They are all we can apply after the infection, right? And here the diseases two types, systematic and non-systematic. And the chemical those act by entering into the system of the plant known as systematic. All therapeutic chemicals given, whereas the other chemicals which do not act systematically are called non-systematic. Some example, protectant chemicals. Here, the systematic chemicals used for systematic disease and non-systematic disease used to non-systematic chemicals. Before that, what all you should make sure about the systematic disease, non-systematic disease. Systematic disease or non-systematic, nothing but localized disease. Systematic disease means the infection occurs the entire the plant body from root to the leaf tip, including all plant parts. That is systematic. After getting infection, we applied therapeutic chemicals and localized. The localized means in particular area of the plant, maybe it confined to leaves or confined to only for the fruits and the root and shoot. This is actually this type of disease. For this type of disease, actually we apply the protector. That means prior infection of the disease, these chemicals we apply. So these are the two types, systematic and non-systematic. And some seed treatment with chemicals can be given here. And the first of all, rotary seed dresser. Let's just wait. Right. So seed treatment chemicals, first of all, rotary seed dresser. Rotary seed dresser. Second one is slurry treater. South seed dip method. The name given dipping. You should understand in this heading. Seed sieving botanicals and hopper box treatment, vacuum infiltration, osmotic priming, salt matrix priming. Actually, here they given 8. But in no need to write all, they just write the some of the important all. It is the important one, rotary seed dresser important, commonly used this one, and slurry treater important, and seed dip method also important. And next actually in this treatment we can use the treatment involved by physical factors also. Here the next was given the page number 46, some of the involving physical factor. In examination, if they ask seed treatment chemicals, in seed treatment process, not chemicals, seed treatment process, you just write some of the chemicals and with help of the physical factors also we should include. Now let's see some of the chemicals instrument here given. First of all, rotary seed dresser. Rotary seed dresser, seed can be the small quantity of seed is treated by the rotary seed dresser. Only this less amount, small quantity of seeds. And second one, slurry treater in given page number 446.1, we can see the diagram also, slurry treater. And it is unlike rotary seeder, in this treater, we can use large quantity of seed. Processing plants is treating by slurry treater. That is the instrument. But in this instrument, they required the quantity of chemicals also. That means this chemical, the produced treater itself. So the required quantity of the chemicals is automatically mixed with specified quantity. That is based on the seed quantity, the some of the chemical released in this instrument. Before the bagging, it should the processing, it should treatment. That means if we large quantity of seed placed in the slurry treater, immediately as such the seeds, the chemicals released and processed. That means the remove if any pathogen, if any untoward parts, they remove it. After that, we bagging that. In a seed processing plant is continuous process and more efficient than the rotary seed dresser. And another one is seed dip method. In name itself given seed dip method, and we can see the page number 444 and diagram 6.2. It involves dipping seeds, propagating material in chemicals. Chemicals, for example, here we can see in diagram also. Maybe here the fungicide. Let's change color. Black color. This chemical fungicide and blue color, maybe blue color bactericide, and let's take another one, green color nematocide. For example, nematocide. Actually, what we done in this situation, in this after taking these two different bowls, these are actually done the before sowing the seeds. The seeds soaking with organomercurial, potato scab, diethane, rhodamel. These are the some of the chemicals. For example, here diethane given, that is the fungicide, diethane fungicide. Now the chemicals, sorry, the seeds placed in this chemical soaked. Example, potato lead white example, one of the disease given inside bracket also. And next one, streptomycin sulfate. The streptomycin belongs to the fungicide. Streptomycin chemical belongs to fungicide. So the next, this blue color chemical streptomycin. The same seeds after fungicide treatment and soaked in the bactericide also. And next we soaked in nematocide also, if any nematodes are present. So hot water treatment followed by seed dip method. After these chemicals, after we are using this different type of fungicide and bactericide, and which follows the hot water treatment. That is, seed dip in the streptomycin sulfate has given very effective result against bacterial disease. Already told you, right? So this is the seed dip method. And another method is seed seeping botanicals. The soaking loose matter of seeds, wheat seeds in 3% onion, 4% garlic extract, 32 degrees, and also make sure the temperature should be in 30 degrees, and we allow it to 29 hours, bring down the infection. However, the soaked seeds can't be stored for more than 10 months, right? So this is the seed seeping botanicals. And another one, of course, here the 4 enough for seed treatment, and also given, let's see the some other also, but not important. Hopper box treatment and vacuum infiltration. In first of all, hopper box treatment, the requisite quantity of the chemicals is mixed with hopper box, the seed drill before the sowing. Simple. That is a special type of box. Already the box filled with some of the dust chemical. We mix it with the dust chemicals. So before the sowing, we take the seeds and sowing on the soil. So this is the upper box. And also we must have seen some of the seed, the powdery appearance appeared in the surface of the seed. That is done by the hopper box treatment. And second one, vacuum infiltration. In that type of also, soaking of the seeds in captafol or binomol, these both are fungicide. A solution for 7 days with the change solution every day. But this treatment's continuation 7 days, with changing the solution for every day. That is one method as a vacuum infiltration treatment, keeping the seed vacuum pump. And the same process the carried out inside the vacuum pump also, vacuum chamber also. So this is the vacuum infiltration. And second one is osmotic priming. In osmotic priming, we utilize some chemical polyethylene glycol, polyethylene glycol peg, which provides osmotic environment. Yet it provides osmotic environment for what? If the seeds immersed in peg solution, what happens? Already told you this osmotic environment. This osmotic environment for what? That means if any pathogens present in the seed, in osmotic priming, what we do, the pegs we have taken the peg solution, we have to take the peg solution and place it the seeds. If there is any pathogen present in the seed, what happens? Plasmolysis takes place. Plasmolysis. But due to the plasmolysis, what happened? Already we discussed in core paper. Plasmolysis occur in the pathogen. What is plasmolysis? Hello, what is plasmolysis?
it is a removal of water. The cell
Right,
become constrict.
right. Removal of water. Here the removal of water, which water removal? Seed water removal or pathogen water removal? If we seed placed in peg, which water removed? The peg actually high concentrated solution. Here the simple if any pathogen present from the pathogen cell, for example, it infected bacteria. From bacteria, the all cytoplasm, the water content, if any water content present, that should be all entire the cell water content loss should be lost. What happened if the bacteria cell water loss? What happened to bacteria?
They will get destroyed.
Yes, they will get dead and it needs to die. So these are all the treatment methods, but remember it, no need to write all. In examination point 2, you should cover apart from the chemicals, that means chemical treatment, and also in the next page given seed treatment involving physical factors. In physical factors also given seed dip in water, first one, and hot water treatment, hot air treatment. The simple, the name itself given clearly. Seed dip treatment means we soaking the seed plain water at 20 degrees centigrade for 41 hours, reduce the loose matter of infection. Without damage to wheat seed, the soaking in water 25 degree for 41 hours. So what happen if we soaking the inside the water, the based on the temperature and hours, if any pathogen, fungal pathogen should be destroyed. And hot water treatment, the hot water treatment also utilized for some of the diseases. That means the same, the seeds are dipped in specific period time or hot water to particular temperature. And you make sure not to harm the seed embryo. That means the hot water limited does not exceed 60 degrees centigrade. When you follow the hot water treatment, you make sure that hot water should not exceed 60 degrees centigrade, 60 degrees centigrade. And also you follow the proper time also. And even below 60 degree, you apply to the seed and you make sure do not exceed the time period 1 hour above. So these are the main considerations. The first of all, in hot water treatment, temperature should be almost given here the below 60 degree. That means the 50 to 60 enough. And treatment time duration also 30 minutes. What happen if you give the above 60 degrees centigrade, And above the 30 minutes, that means above 30 minutes to 1 hour, what will happen to the seed? If we exceed the temperature and exceed the time duration, what happen?
The seed will get destroyed. It
Yes,
will
seed will
lose
get,
the
how it is
germinating
destroyed?
capacity.
Right. The embryo can survive the below 60 degrees centigrade only. If we exceed the temperature, that temperature with that above 60 degrees centigrade temperature, the embryo should be destroyed, deteriorated. And also we should not continuation time also. 30 minutes enough. Even if you taken 50 degrees centigrade, but in placed in 2 hour, 3 hour, what happened? That period also not perfect to the hot water treatment. And third one, hot air treatment. Hot water treatment, dipped in the water, hot water. In hot air treatment, we expose the hot air to the stream for specific period, which is safe for viability of the seed method, the more effective for the pathogen, if any pathogen present surface. In hot water treatment, mainly hot water treatment, if any,
Sir,
uh,
uh, explain the fifth one, no, sir. The third and fourth one is easy.
okay,
Seed seeping.
right. Fourth one also solar energy treatment also common. That is the traditional method, right?
Yes, sir.
Right. Seed seeping method. In seed seeping method, it 20% commercial salt. The seed seeping, we apply the commercial salt. Salt treatment, simple. Effective, eliminate the lighter, toffee, kernel, bunt, infected seeds, nematode, gall. These are the diseases symptoms. Toffee, kernel, bunt. So these symptoms effectively eradicate with help of this salt treatment. And this method can be partially used to separate the sclerosia erga. Some of the disease, particular disease also given. Ergot of viral, sorghum and relicate. Floated material contains ergot, especially ergot diseases. Ergot disease eradicate through this treatment. And seed seeping treatment, remember, we apply the salt, simple. Apply the salt to the seed. That is the seed seeping treatment. Is it clear?
Yes, sir.
And another easy one is 6.4, treatment with biological agent. How can you understand? What can you understand? Biological agent. How the biological agent treat the seeds? Just assume, simple. First of all, in chemical, we have seen some of the chemicals and physical factor to seed treatment. The biological agents. What is biological agent?
Small microorganisms.
Simple. One organism kills the another. That's it. So in microorganisms have also been successfully used to as a seed treatment. Here the seed treatment with Fusarium moniforms inhibits the kernel infection by Aspergillus flavus. In maize plant, here another treatment of brassica seed with Ilcodium roseum and increased seedling emergence. The several formulation biological agents are available in the market. And another one given here, the Pseudomonas bacteria, which controls some soil-borne diseases. So in this method, we don't apply any chemical, any physical factor. We're using some of the microorganisms, the benefited microorganisms, not harmful. These benefited microorganisms fight with others, that mean harmful microorganisms. As a result, we get pathogen-free seeds. So do you know probacteria, prebacteria, which are useful to our gut in general.
Yes, sir.
In general, some of the in market available to human beings, some of the pre-bacteria, probacteria, or probiotic. What is the purpose of probiotics? Prebiotic and probiotic. How is utilized for human beings?
They will keep the person healthy
How they keep the person healthy?
by providing good bacteria.
Providing good bacteria and eradicate the bad one also.
Yes.
Right. That's why the most of the people suggested to uh, we take care the curd in every in our lunch and dinner. In curd there are several probacteria, prebacteria are there. That's why frequently our elders also frequently told that one. And next one, is it clear? The chemicals, these are clear.
Yes, sir.
And next one, and here the polymer coating and special method of application. It is one type of the chemical method actually. The coating of what happened here in this, the coating of seed surface with specific polymer, such as on God, as it increase the efficacy of the fungicide to control seedborne disease of maize, especially in maize. Actually, we apply this polymer coating before infection, not after. Before infection, if we prior to given this polymer coating, there is no chance to infect the fungal pathogen, right? And the treatment of seeds with carboxyl fungicides. So these are the other chemicals. We apply the as a coat, the other chemicals also we can use, right? So anyway, in polymer coating specialized method, we extra coating with help of the some of the chemical prior to the infection. That is most important one, right? Is it clear? Sixth topic clear? Hello?
Yes, sir.
Right. In this, the most important questions are in sixth brief, explain the seed treatment with chemicals. And the second one, seed treatment involving the physical factor. Actually, the physical factor only, that is not long question, the short answer that also. That the only one and only essay one, the briefly explain seed treatment with chemicals, one question, and briefly explain about the seed treatment, second one. If they ask about briefly explain the seed treatment, we can write about the chemicals, about the physical factors, and about the treatment with biological agent, and again the polymer coating. They are all. If you learn the entire long question, they question repeated in short answer also. And given page number 48, given second weight, the seed dip method, hot water treatment, solar energy treatment, treatment biological agents, these are all comes in. It's a long question. But
Yes,
again,
sir.
they are repeated separately for short answers. Right?
Yes, sir.
Okay? Right. So this unit also very easy and important. Already told you, just focus on any two blocks, complete two blocks. After that, you select the any units. For example, the third block and second block easy. You select the third and second block, and you should select the important question from remaining units, remaining blocks. That is your wish. And another one, block three. Block three also important. The structure of pollen and ovule and seed development. First, so let's see the seventh unit. The structure of pollen and ovule. Already you must have learnt in the fourth semester embryology about the pollen and about the ovules. Anyway, once we recollected that one, that once again, uh, Let's see the structure of pollen grain.
Sir, in 20 marks question, which question we can expect?
In 20 marks, generally we have to ask outcome based. Outcome based means, for example, so far we completed, for example, what we completed so far: seed viability, genetic erosion. It is not, there is not chance. Here the seed marketing. Seed viability, okay. Seed marketing. The fifth unit, seed marketing. In seed marketing, generally we can ask how to marketing for promotional of the seed. As a that means as a salesman, how can you promote the seeds? There is just that type of question. What would you write in that situation? How can you promote? How can you promote?
By
Hello?
electronic media, social media, by the newspaper.
Right, that's it. That's it. And another type of question. Uh, Seed storage, low temperature, no chance. Dormancy, no chance. Standard. Seed degradation mechanism, no need. Symptoms. And there is a chance to ask continuation question. Continuation question means that means the seed marketing promotion one question and continuation. Tell me some of the seed treatment, the olden days seed treatment process. Olden days seed treatment process. What are the olden days seed treatment process? In previous days,
Solar,
farmers how to treat the seeds. Can they
solar
utilize the
energy.
chemicals in olden days? Can they utilize the chemicals? Can they utilize
No,
the slurry treater and rotary seed dresser, seed dip method,
no, no.
osmotic priming? They cannot use, but only just almost the physical factor, all physical factor they can apply. Mainly hot water treatment, and especially the most important one is solar energy treatment. That's it. And here given in block 3, structure of pollen and ovule. That is direct question. No need, no doubt at all. And next one, 4 seed testing. So almost entire seed technology cover the outcome based questions. And maybe they ask in about write about seed storage process. Why we why we store the seeds? Why should we pack in the seed? And write about the storage types. They are outcome based. They are outcome based. Of course, in this, any type of question comes from the book, but indirectly, right?
Yes,
Next
sir.
one,
And
right? And here the structure of pollen grain and ovule. First, let's see the pollen grain structure. Pollen grain. The pollen grains, microscopic, they are in different in size and shape. All are same in size, all are same in shape. And how many cells? Microspores or pollen grain made up of how many cells? Actually not given here, but remember how many cells? One pollen grain or one microspore. Don't get confused, both are same. Pollen grain, microspore, both are same. Tell me.
2 cells.
Here how many cells?
It is given cell. 2 cells they are made up of.
Where they given?
On structure of pollen grain in the 7.2.
In 7.2, where it is? Pollen grains are microscopic, different in size and shape, tiny herbs, while others are
Second,
egg-shaped. Second para, through the microspore, pollen grain hardly looks real, an extremely durable body. It has tough outer coating. This hard coat offers great protection from the harsh outdoor environment. This is important because inside this tough cell in 2 cells. The important because inside this tough cell lie 2 cells. The tube cell and generative cell. What happened here? Actually, the microspores are pollen grains, not two-celled one. Remember it. Actually, the one word missing here, the two-celled structure, not a microspore, not a pollen grain, but that is male gametophyte. I will explain clearly. First of all, remember it. Pollen grains are microscopic, okay. Different size, okay. And also some shapes also given. That is another matter. But this microspore grain, it is made up of, this pollen grain made up of 2 layers. 2 layers. Single cell, unicell, only this is one cell, unicell, remember. Microspore pollen grain, unicell, and it covered by 2 cell. Already you know, outer one, exile, intile, exile and intile, and intile. And on nucleus, let's see the color. The nucleus. Right? So this is actually pollen grain. But what happened? Remember, please remember it. The pollen grain or microspore further developed into male gametophyte. While it is developed into male gametophyte, this single cell nucleus I will draw in this also. In single cell nucleus divided into two. Divided. Two nuclei formed. And later these two nuclei develop into 2 cells. Septiform, 2 cell. Tube cell and generative cell. One is tube cell, this one, and generative cell. Actually, this 2 cell structure not a pollen grain, not a microspore. This 2 cell structure called as male gametophyte. Remember it. Write down there. Actually, the important because inside this tough shell in place of the tough cell, inside this male gametophyte. Actually, before that, you should write the microspore further developed into male gametophyte. A continuation, the male gametophyte made up of 2 cells. One is tube cell, another is generative. That's it. Is it clear? Hello?
Yes, sir. Sir, repeat once again. No?
First of all, the structure of pollen grain clear?
Yes, sir.
Simple. The pollen grain, microscopic, variation shape and size, unicell, remember once again, uninucleus, unicell and uninucleus covered by 2 layers. outer exile, intine, right? But actually, this pollen grain developed into male gametophyte. When it is developed into male gametophyte, after the one nucleus divided into two nuclei, and finally developed into these two nuclei surrounded by cytoplasm and septa form become into two cells. Now this is called as male gametophyte. In male gametophyte, two cells are there. One is tube cell, second one is generative cell. Right? Is it clear?
Yes, sir.
So don't get confusing here. Where is male gametophyte? Where is male gametophyte? Development of male gametophyte. Where it is location? Hello?
Nucleus
Where is the nucleus?
in the middle.
That part belongs to
Pollen grain.
that's it. That's it. Inside the pollen grain, male gametophyte present. That's simple. Inside the pollen grain, male gametophyte present. That already the single layer, first one single cell, two layered, but it becomes two. But the two cells present inside the spore. Inside the spore. Right? So the male gametophyte developed inside the microspore. Is it clear? And the another important one is the chemicals. The outer layer exin chemical, which is chemically made up of sporopollenin. And the chemical also here, exin chemical, sporopollenin. And entire chemical pectocellulose. Only here given cellulose, I think where it is. Where is entire? Where is entire? But remember, the thick, highly and complex outer layer rich in compound known sporopollenin, waterproof resistance, stiff, okay, advanced polymer, okay, in addition. But intent, remember, intense chemical pectocellulose, pectocellulose, right? Is it clear?
Yes, sir.
Right. So this is about the pollen grain structure, and the structure also given in page number 5.52, figure number 7.1. Right? Is it okay? So this is also short answer. Structure of pollen grain short answer. And another one is structure and types of ovule. Ovule, another word, megasporangia. First, let's see the structure. After we discuss about the types of ovule. These two important, especially the types of ovule, very, very important for short. Types of ovule, very, very important. Already you must have learned during fourth semester and second year. Okay? Right. Let's discuss here. The first, let's see the structure. Megasporangium or ovule, the mainly two parts. Let me draw just rough diagram for our easy understanding. Sorry. Integument. These are the integument. And another one is, let's change the color. Nucellar. The green color nucellar. Only two parts you can see here. Integument nucellar. So another part here, the ovule is considered to be the integumented megasporidium. That's why it is called as integumented megasporangium. The ovule is consist of the stalk and the body. This is stalk. Sorry, this is stalk, and this is body. Stalk and body. The stalk is called as funicle. The stalk name is called as funicle. Please remember this word, funicle. Where is funicle? Where is body? Because these are important for types of ovule. And one end of the funicle is attached to the placenta. What is placenta? What is placenta? And other end attached to the ovule. So this is attached to the ovule here. So this is ovule body. This is placenta. Placenta ovary part. In ovary part, actually one end attached to the placenta, other end attached to the ovary body. The point of attachment of funicle with the body is called hilum. Where it is attached, that is called hilum. That is called hilum. Sometimes funicle get fused with the body of the ovule one side and forms the ridges known as raphe. So sometimes what happens if the stalk deeply attached with the body, there is a formation of raphe. It is sometimes occurs in some ovules only. The body of the ovule shows two ends. The basal end, often called the chalaja, this end, basal end, this end. This is also very important. Basal end called as chalaja or kalaja, and top end, this is called as micropylar end. Micropylar end. The main body of the ovule is covered with two envelope. Already told you, two outer, inner, two integument. This leaf opening the top ovule called micropyle. This place called as micropyle. Already told you. The integuments enclose the large parenchyma tissue known as nucellus. Already at the very beginning I told you. The ovule made up of mainly two parts such as integument, this red color, and green color nucellus. That's it. Now here given the different and the residual part of the nucellus in the mature seed called perisperm. The center of the nucellus is situated in the female gametophyte. I will explain later about how the formation of female gametophyte, like a male gametophyte, right? So here the one word is perisperm. Simple. Perisperm means after fertilization, actually the nucellus disappear. It was to disappear, but even after fertilization, if any nucellus present, that is called perisperm. From the nucellar cells, remember this green color nucellar cell. From the nucellar cells, one of the nucellar cell act as a megaspore mother cell. Just remember, don't write. Megaspore mother cell. This megaspore mother cells further developed into megaspore. This megaspore further developed into let's change color, red color, female gametophyte. Female gametophyte. Where is female gametophyte now? Arun? Where is female gametophyte? Or female gametophyte or embryo sac? Rather than embryo sac, where it is?
The ovule.
Inside the ovule, that's it. How it is developed? From which part? What are the green color?
Green color is the nucellus.
Yes, right. Nucellar cells. There is a process. First, nucellar cell develop into megaspore mother cell. Megaspore undergoes to meiosis. After meiosis, megaspores are formed. Now this megaspore developed into embryo cell. And here the in development also monosporic, bisporic, tetrasporic are there. Monosporic involved 1 megaspore. Bisporic embryo sac or female gametophyte development by the help of 2 megaspore. In tetrasporic, the development of embryo sac or female gametophyte by 4 megaspore. That is clearly given in previous year, 4th semester, but here no need. Just remember the embryo sac or female gametophyte developed inside the ovule. That's much enough. And also based on integument, they are classified into unitegmic, bitegmic, and extra one called as aryl and carancule. Ategmic. First of all, let's complete about ategmic. A for absent. In the type of ovule, there is no tegma. Example, parasitic like Loranthus viscum, Santala. Unitegmic, to name itself given uni. What is uni? What is uni?
One.
One. Made up of none rather than two. Only one is present.
Sir,
Example,
in
simple.
bio also only one is there.
Where?
In biotechnic,
In biotechnic.
it should be two, na, sir?
Yes, at 100% two. Actually, it is wrong given. Biotechnic ovule with, of course, actually, integument with two layer. That is the correct one. Integument, single integument with two layer. First one, unitegmic, single integument with one layer. Integument, one layer. Bitegmic, write down the ovule with single integument with two layer. Integument may be developed by the single layer or may be developed by the double layer. Okay?
Yes,
Just
sir.
add it. In bitegmate, ovule spelling also wrongly mentioned here. Just
Yes, sir.
correct it. The ovule with single integument. Example, sympatale, gamma petale. Single integument with two layer. Add it with two layer.
Sir, in both the examples are same.
No. Oh, sorry. Yes. Actually, unitegmic, write down here unitegmic. Sympetale, of course, maybe gamipetale, but not a dicotyledon, monocotyledon. Instead of dicotyledons, monocotyledon, write down monocotyledon, unitegmic example. Unitegmic example, monocotyledons. Have you changed?
Yes, sir.
Right, bitegmic, the same. Bitegmic, no change. Sympetale, maybe sympetale or
Dichotomy.
gamipetale, dicotyledons also. Right. Aryl. Aryl, this is the extra layer. Third layer, the considered third layer. In aryl one, integument must be two layered, no doubt at all. And extra layer also present around the integument. This is mainly found in lychee, nutmeg. The best example we can see. Have you hear the word simachinta?
No, sir.
Pitkalobium?
No, sir.
Pitkalobium also there. What can I say? Just type in Google Pitkalobium or simachinta, the better one, simachinta.
Sir, can you tell the spelling?
Do one thing. The example here, integument, where it is. Lychee. No, actually, just wait. Lychee also not correct. Simachinta. Right. SEEMA Simachinta. CHINTHA.
Seema Chintakaya, I think.
Yes, yes. Type it.
Yes,
Type
sir, I
enter.
type.
Did you any image get it?
Yes, sir, it's like a what we call imli, like
Right, right,
pods are there.
right. If you remove the outer pericarp, what you observe, the inside the black color seed we can observe, right? What is
Yes, sir.
present the above the seed? Actually, these are the common in villages.
Okay.
What
Some
is
white
present?
color top.
Right, white color. The white color part called as erin. White color part called as erin.
Okay.
Right, and also add the another example for the eril. Only lychee, nutmeg. Apart from that, the best known example, the common example is Pithecanthropum. Write down Pithecanthropum. That because it is very common. Type in Google and correct it. Pithecanthropum.
P-A-T-L-O-B-I-U-M.
Yes, Dulce. Dulce. English name
Yes.
Madras thorn. English name Madras thorn. This is the scientific name Pithecanthropum dulce. Scientific name Madras thorn. English name. Okay.
Yes, sir.
Right, and next one is Karankul. Do you know